Welcome to the problem-solving set on the Central Dogma translation. Problem 1. A nonsense-suppressing tRNA is one that brings an amino acid to a nonsense codon, suppressing the mutation and allowing for elongation. One such tRNA is known to suppress more than one nonsense codon. Determine its anticodon sequence, which stop codons it can suppress, and what amino acid it is likely to carry. So there's some theory that we have to go over before we can solve this problem. First, a nonsense codon comes from a nonsense mutation, and this encodes for a premature stop. However, it's saying that these nonsense-suppressing tRNAs, instead of signaling for translation to stop and the ribosome subunits to come apart, it's actually bringing down an amino acid to incorporate into the polypeptide chain, and so elongation can continue. So the stop codons that we know of are the three regular stop codons. So these are UAA, UAG, or UGA, so that's the first piece of theory. The other theory we have to go over is the wobble theory, and we're going to use these wobble rules to solve this problem. So the wobble theory, this is where in a codon that runs 5' to 3', right? So we have a codon running 5' to 3'. Now the wobble theory states that the first two nucleotides in a codon are more important for binding base pairing with the anticodon than the third base is. So we could say 1, 2, 3, and we'll say that this nucleotide is not as important for base pairing. Now in terms of anticodon, remember that anticodons run antiparallel to codons, so they run 3' to 5'. And this means that the base pairing here, it is the 5' nucleotide in the anticodon that can not be perfect. It doesn't have to be a perfect match. So just keep that in mind as we solve this problem. So again, we have these three stop codons that could possibly be our nonsense codon. Which of the two, it's asking, which stop codons can it suppress? So if you look, these first two both start with UA. Remember again that the first two nucleotides are important for base pairing, but that the third one, there's a little slack. It can be slightly different. So what we would expect is that these two codons are actually the ones that are suppressed by this one tRNA. So we can write, we'll just cross this one out. And again, that's because the second nucleotide is a G, so remember the first two can't be different, they have to be the same. So we'll cross that one out. These are the two stop codons that this tRNA can suppress. So write that. And now it's saying to determine the anticodon sequence of this suppressor tRNA. Well, if the codon is either 5' UAA or 5' UAG, the anticodon that will suppress both of these is going to be 3' A to pair with the U to pair with the second A, and remember that this is the one, we'll write W for wobble, is the one that has some slack. So we're going to write a dash here and 5', and that's because, again, the third nucleotide in the codon or the 5' nucleotide in the anticodon is the one that can be different and still base pair and bring in an amino acid. So this is the anticodon of the suppressor, 3' AU dash. Finally, the question is asking which amino acid is it likely to carry? So the easiest way to look at this is to look at it from the codon point of view. So if you switch around the anticodon and meet it with a codon, you'll get 5' U A dash. Now this makes sense because the two stop codons fit into that category. It's either UAA or UAG. But if you look at the genetic code chart, UA dash could also mean UAU or UAC. Now both of those codons encode for the amino acid tyrosine. So for the amino acid that it's likely to carry, we're going to guess tyrosine. And that's because the anticodon on the tRNA carrying tyrosine with the wobble rules can actually base pair with these stop codons and suppress the nonsense mutation. Problem 2. According to the wobble rules for pairing, inosine I in the anticodon can pair with U, C, or A in the 3' position of the codon. Of the following amino acids, which is most likely to be translated with a tRNA having inosine in the 5' position of the anticodon? So there's 5 amino acids here, and they're actually going to ask us about them again in Part B. So why don't we go ahead and write them out. So we have proline, histidine, methionine, cysteine, and tyrosine. And we're going to take a look at our genetic code chart and see what are the normal codons that encode for these amino acids. So proline has 4 different codons. So we're going to write that as CC dash because the last can be any of the 4. For histidine, we have CAU or CAC. For methionine, there's only one, and it's the AUG. For cysteine, we have 2, so it's either UGU or UGC. And finally, for tyrosine, there are 2 as well, so it's either UAU or UAC. So it's saying, out of these 5 amino acids, which is the most likely to be translated with a tRNA having the inosine in the 5' position of the anticodon? So remember that these that we just wrote down, these are codons. And these run from the 5' to 3' position. And anticodons, from left to right, run from the 3' to 5' position. Remember, because you have to do complementary base pairing, just like we've seen in regular RNA, in DNA, everything. So it has to be anti-parallel, and the anticodon has to be going from 3' to 5' to match up with the codon. So it's saying which one can have an inosine in the 5' position of the anticodon. So it would be here. And if you notice, that matches up with the 3' position of the codon. So this is the one that we're concerned with. So it's basically saying that that question mark can either be a U, a C, or an A, because that's what it said that I pairs with. Now, if you look at proline, proline can either be in the 3' position, can have a U, an A, C, or a G. So 3 out of its 4 possible codons could bind with it. For histidine, the U or the C can both bind with inosine, so these are 2 codons that have it. So for proline, let me just write 3 instead of 3 out of 4. For methionine, it's a G, and that can't bind with inosine, so that's 0. Cysteine has a U or a C, so there's 2 codons that can bind with the inosine. And tyrosine, again, is U and C. So the one that's most likely to be translated with the tRNA having this in the position of the anticodon is proline. And that's because 3 of the possible codons that encode for proline can bind with this altered base. Now for Part B, it says of the same amino acids which cannot have a corresponding tRNA with I at its 5' anticodon position. Now here we already said it was methionine. And that's because the 3' position of its codon is a G, and G is the only base that cannot bind with this inosine in the anticodon. So methionine is the answer for Part B. Question 3. If an RNA molecule has 72 nucleotides, what is the maximum number of amino acids that it can encode? But remember that amino acids are encoded by what is known as codons. And codons have 3 nucleotides, right? So you could just take out a calculator and do 72 divided by 3, and that gives you 24. So you might want to say that it can encode for 24 amino acids. Now, if this was an RNA molecule that was part of the polypeptide that's on the end, the answer isn't actually 24. So if the protein was, this is the total protein, or it's a piece from the protein at its end, then you could actually only encode for 23 amino acids, because one of those codons, the 3 nucleotide group, has to be a stop codon. Now remember that stop codons don't encode for any amino acid specifically, it's just to release the ribosome and to stop translation. However, if this was in the middle of a protein, polypeptide, then you can encode for the full 24 amino acids, because the stop codon will come later on in the polypeptide. So if your professor asks you these types of questions, just make sure you know before the test if he's going to specify if it's in the middle of a protein or it's the end or total protein. Question 4. How many amino acids are coded for by the following coding DNA strand? So you want to take notice that they're giving you the coding strand. Now remember that the coding strand is analogous to the mRNA that's going to be made, except that all of the T's are going to be changed to U's for uracil. So first let's write what our mRNA molecule is going to be. So it's going to be in the same polarity, start with 5', and all the T's are going to be changed to U's, while all the other nucleotides are going to stay the same. So I'm just going to copy this over, changing all the T's to U's. Okay, and then remember you're going to end with the 3'. So now, in order to find out how many amino acids we're coding for, we're going to look for 2 special things. The first is that we're going to look for a start codon. Now the start codon runs 5' to 3', and if you look at the genetic code chart, or if you just remember it, the start codon is AUG. We're also going to try to see if we see any stop codons. Now there are 3 possible stop codons, and they all run from 5' to 3'. And they are UAA, UGA, or UAG. Okay, so any of those 3 will signal the ribosome to break apart, the subunits will break apart, and translation will end. So first we're going to look and see, running from 5' to 3', where is our start codon? So if you see U, U, G, that's not one. And then the next codon we see AUG, this is the start codon. So that's going to be the amino acid methionine. Then this next codon is going to give us another amino acid. Then we have another. And then, if you see, this next codon is UGA. Now that is a stop codon. Stop codons do not translate with any amino acid. It's just a signal for the ribosome to release. So although it doesn't say it here, so it's asking us how many amino acids there are, we have the start, which is methionine, and then you have 2 more codons to give you an amino acid before you hit the stop. So total, this will be 3 amino acids. And it doesn't ask for us here, but you might as well use the genetic code chart and see what amino acids these are. So we know it's going to be methionine first, because that's what the start codon encodes for. Then we have CCU. So if you look over CCU, it's going to give you proline. And then we have GAA. So GAA is going to give you glutamate. Again, remember, it's up to your professor if you have to remember the 3-letter or 1-letter abbreviations for these amino acids, but remember that the GLU is for glutamate, whereas the GLN is for glutamine. So this is going to be the really short polypeptide that is translated from this mRNA sequence. Problem 5. A peptide has an amino acid sequence that begins methionine, histidine, lysine, alanine. If the amino acid tRNA with the anticodon 5' GUG 3' is altered so that it now carries lysine, what would the altered peptide begin with? Well, remember that anticodons, the way that we normally read them, are from 3' to 5'. So this is palindromic, so it doesn't really matter in this case, but you always want to remember that the anticodon, if they give it to you in this way, 5' to 3', to just switch it around. So it's going to be 3' GUG, and this will complementary base pair with the codon 5' CAC. So this is for the tRNA that was mutated, where the anticodon wasn't changed, but the amino acid that the tRNA is carrying is changed. So the anticodon is going to stay the same, but now this one is going to carry lysine. So what we're going to do is look for what this codon would normally encode for, and then whatever that amino acid is, that's going to be changed to lysine. So if we look at the genetic code chart, we're looking for codon CAC. So if you go over to CAC, this normally encodes for histidine. So we'll say normally encodes for histidine. But now, because the amino acid tRNA was mutated that it now carries lysine, any histidine in this polypeptide chain is now going to be replaced with a lysine. So this polypeptide is going to begin methionine, lysine, lysine, alanine. So just to go over that again, the sequence began methionine, histidine, right? So that second codon in the mRNA would normally encode for histidine, which is the codon right here. That codon normally encodes for histidine, but the tRNA that it is base pairing with has the same anticodon, but is now carrying a different amino acid due to a mutation. So instead of this codon encoding for histidine in this case, it would now bring down the amino acid lysine.